Okay, so we're starting out with the general anatomy of the nervous system. And just to set some context here, this unit is really going over the gross anatomy of the nervous system. You might have talked about some of these structures in your lab already, the meninges of the central nervous system of the brain and spinal cord. We're going to talk about gray matter, white matter, the types of cells that are found in the central nervous system. So neurons as well as the supporting cells. So this is going to be sort of an overview as we move into discussing the two divisions of the nervous system. So we'll talk generally about gross anatomical features and functional details of the nervous system. And then our next unit gets into the autonomic division, followed by the somatic division. So we're kind of zooming out from where we were in module 2 when we were talking about more of the cellular details, more of the organelles, the parts of the neuron, the details of the action potential. So we're going to really kind of zoom out from that and take a more holistic look at the nervous system as a whole. So our first objective is to talk about the different types of neurons, as well as their supporting cells, and then the functions of each. Okay, so let's think about the central nervous system, which is the brain and the spinal cord. And that can be compared or contrasted to the peripheral nervous system, which again, we'll get into coming up. This is going to be the peripheral nerves, the ganglia, etc. So when you think about the CNS, it's essential for maintaining homeostasis. We've talked about the homeostasis loop and the way that information is being received into the CNS on afferent fibers. This is bringing in instructions about our set points, our regulated variables, your blood pressure, temperature, things of that nature. That type of information, sensory information, is going to be coming up to the central nervous system. All right, whether that's brain or spinal cord. And then instructions are given to respond to those stimuli. So for example, if it's a blood pressure drop, the instructions that are sent would increase heart rate, and that would correct the drop in blood pressure. So the central nervous system is crucial for maintaining homeostasis, right? It's kind of the CEO, taking the stimulatory or sensory information and then giving the corresponding responses. It has about 100 billion neurons forming 100 trillion or so connections. So a lot of interconnectivity between the central nervous system formed by synapses across neuron to neurons, right? We've talked about those types of connections. This is responsible for everything that we perceive, do, think, feel, and not just in a practical sense, not just the words or thoughts, but really in our personality, right? Our memories, our reactions, certain aspects of our personality, right? These are all being controlled and governed by our central nervous system. Okay, so pretty much everything about that defines who you are and separates you from another person is essentially governed by the CNS. It's also one of the least understood organ systems. So while there's so much complexity, there's a lot that we still don't quite fully understand. And so we want to think about this as just really the surface layer of the central nervous system. There's a lot of depth and complexity that's still being discovered and investigated, right? Very, very complex system. Now let's look at the players in the central nervous system. First, we have neurons, and neurons are excitable cells. These are the cells that have the ability to fire action potentials and to transmit those action potentials across from one to the next, right? So neurons are excitable cells in the CNS. Glial cells, on the other hand, and we'll talk about the different types. There are 5 different types of glial cells. They are non-excitable. They are supporting cells. They cannot necessarily fire action potentials, but they can receive information from neurons in other ways. They can be involved in cleaning up debris, any dead neurons. They can be involved in clearing cellular debris away and helping to regulate the environment that the neurons are in. We're also going to talk about the physical support, so the meninges that actually help to cover the brain and spinal cord. Then we'll talk about the blood supply and some of the defects, how we can get into strokes when the blood supply is interrupted. Then we'll talk about the blood-brain barrier and then gray matter versus white matter, right? Myelination versus unmyelination. Okay, let's begin by looking at a neuron. And a neuron is the structural and functional unit of the nervous system. Okay, this is the level at which all of the functional interactions in the CNS actually happen from one neuron to the next. They are responsible for responding to chemical and physical stimuli. They can conduct electrochemical impulses. We've talked about these. These are called action potentials or graded potentials, right? They can be involved in releasing chemical regulators. These are neurotransmitters, acetylcholine or norepinephrine. These regulators help to send these signals as chemicals from one synapse, one neuron to the other. They are responsible for our perception of all types of stimuli, right? Whether that's physical stimuli, chemical stimuli, olfaction, sight, hearing, touch, taste, all of those stimuli, which include things like learning, formation of memories, controlling of our muscles, glands, adipose, all of those are under the control of neurons. All right. Lastly, neurons cannot divide, but they can repair. So this is why when you have a spinal cord injury, a peripheral nerve injury, let's say you have a slipped disc that actually impinges one of your sciatic nerve, for example, that causes pain and numbness to the legs, those things are really difficult to heal or repair because the cells cannot just divide and create new ones, but they can repair, which takes a lot more time, physical therapy, and that sort of thing to begin regenerating those neurons, right? Very difficult to reproduce, but easier to repair just over time. Now we can classify neurons based on their structure, the way that they look. So the number of processes, axon terminals that are extending from the soma or the cell body. So we can structurally divide neurons depending on whether they're unipolar, pseudopolar, multipolar, how many processes are extending from the cell body. Now, the more meaningful way to classify neurons is functionally. Okay, functionally, we can classify neurons based upon what type of stimulus or what type of impulse, rather, they're transmitting. Is it a sensory impulse? Are they receiving information from the world around us, sight, smell, touch, and sending that to the nervous system? Or is it a motor neuron? Are they transmitting motor impulses? Are they giving instructions to skeletal muscle, right? Or to cardiac muscle or smooth muscle? Or they can be interneurons. They're going to be involved in both and really just helping to transmit that signal from one to the other. So this is kind of just mixed. So that's the more meaningful way, and that's the way we're going to focus on in terms of classifying neurons based upon their function. What type of impulse are they transmitting? So the functional classifications, we can kind of dive into this further. Sensory neurons are going to conduct impulses from sensory receptors, again, out in our peripheral nervous system, up into the central nervous system, starting at the spinal cord, but going all the way up to the brain. So this is going to be generating impulses in the afferent direction, right? From the world into the CNS. That's going to be afferent with an A. Motor neurons, on the other end, are going to be conducting impulses in the opposite direction. So from the CNS from the CNS, spinal cord or brain, out to skeletal muscles. Okay, out to skeletal muscles. And if we think about motor neurons, this is going to be going from, or we can further divide motor neurons rather into somatic impulses going to skeletal muscles. This is under our voluntary control. So this is going to the biceps, the triceps, the glutes, right? Muscles that we can voluntarily regulate. The other division of motor neurons are autonomic motor neurons. Same direction of impulses, but this is going to different types of muscle. Smooth muscle, cardiac muscle, glands. These are structures that we do not have voluntary control over. So the autonomic motor neurons, we discussed this a bit earlier, these are going to be less myelinated, right? Because the impulses are going to be traveling slower versus somatic neurons are going to be heavily myelinated because these skeletal muscles need to be more responsive, right, to our control. And so there's going to be a faster velocity of the action potentials on the somatic side of the motor neuron. And then association or interneurons are pretty much connecting the two. So if you look at the association neuron, it's going from one area in the spinal cord to another area in the spinal cord, basically bridging the gap. And they can play a part in transmitting both sensory and motor impulses. Let's move to the other types of cells. So we've talked about the neurons. Let's talk about their supporting cells, which are glial cells. Now, this is going to make up about 75 to 90% of the central nervous system. And together, this is collectively known as neuroglia, right? A collective substance called neuroglia. And if we look at the different types of cells, there are 5, which we'll talk about individually, but they're all non-excitable. As I mentioned earlier, they do not transmit action potentials or electrical impulses. They're really just playing a background supporting role. Think about them as like being the background singers or the background actors, really just supporting the star of the show, which are the neurons, right? The excitable cells. Now we can see all of the different types here. We're going to go over them individually, but they have different locations and different functions in terms of helping to support the neurons, which is here. This is one of the major neurons. But then all of these cells here with these star-like processes, these cells lining the cavities, these cells lining the blood vessels, all of these are under neuroglia. So here are the 5 cell types. Astrocytes. Astrocytes are these star-shaped cells. That's how they get their name. They have many foot processes sticking off, some connecting to neurons, some connecting to the myelinating cells, which are Schwann cells here, some oligodendrocytes here, some connecting to the blood vessels, and then some connecting into the ventricular system. So astrocytes, because of their multiple foot processes, they're going to be helping to monitor and surveil different parts of the central nervous system. Ependymal cells are going to be lining the ventricular cavities. So these ventricular cavities within the central nervous system are actually housing cerebrospinal fluid, or CSF. And that CSF is produced by these ependymal cells. So these cells not only line the ventricular cavities, but they also make the CSF that is circulating in those cavities. Microglia or phagocytes, these are cells. So here's one down here, here's another. These are cells that are also having sort of several processes sticking out, but the role of these cells is to engulf and destroy any foreign pathogens, bacteria, viruses, cellular debris. These cells are going to be playing sort of an immune role in removing any threats or toxins or pathogens in the CNS. And then the final 2 cells are going to be myelinating cells. Oligodendrocytes are going to be cells that myelinate in the central nervous system. So here are a few oligodendrocytes, and you can see them actually extending their foot processes to make the myelin sheath that actually myelinates the axons of the neurons. Okay, this is how we have the nodes of Ranvier and saltatory conduction. That is because of the myelin that we've talked about. So the cells that actually lay down that myelin are oligodendrocytes in the CNS. And then out here in the PNS, the cells that myelinate are Schwann cells. So Schwann cells will myelinate peripheral nerves, anything that's outside in the peripheral nervous system, and oligodendrocytes myelinate cells and neurons in the CNS. So here are astrocytes. Again, they have a star-shaped appearance, right, with their many foot processes. Their connections are to extend between multiple structures, right, between nervous tissue, such as the neuron, and non-nervous tissue, such as the ventricles, the blood barrier. So their foot processes are going to be helping to connect multiple structures. Their foot processes are also going to be forming the blood-brain barrier. And the blood-brain barrier is what protects the central nervous system from anything that could be circulating in the blood, right? Let's say you had a viral infection and you have viruses circulating in your plasma, right, in your blood. What prevents those viruses from escaping and actually attacking the central nervous system is the blood-brain barrier. And we'll talk about the junctions that actually form that barrier, but astrocytes and their foot processes spanning the capillaries of the central nervous system are what help to form, that barrier. Another function that astrocytes have is for modulating synaptic activity. So if you recall us speaking about presynaptic facilitation, presynaptic inhibition, where we had these foot processes modulating the presynaptic terminal, astrocytes can play that role as well. This is another way to recall that they're not excitable. So they're not going to directly encourage an action potential, but what they do is encourage more neurotransmitter release from the presynaptic terminal, and then that can influence the greater potential that is initiated on the postsynaptic side. So astrocytes can help with presynaptic modulation as well. Okay. And then lastly, just to communicate, sending chemical messages, sending responses based upon the needs of the neurons, as well as just communicating between all of the different types of glial cells. Okay, another function here, removing neurotransmitters. If there's excess neurotransmitter at the synaptic cleft, astrocytes can help to clear that away from the junction as well, especially glutamate. Glutamate can be highly toxic because it is stimulatory. So it can actually result in spasms, right? Continual contractions, just overall overexcitation. And so the astrocyte can help to clear excess glutamate. Also just helping to maintain electrolyte balance of the interstitial fluid. So making sure that the right amount of sodium, potassium, chloride, calcium exists in the interstitial fluid that is bathing the central nervous system and the neurons. And in doing so, it also protects against oxidative stress, toxic stress as well. And then another function is glycogen synthesis. We will speak about glycogen and glucose when we look at the endocrine system coming up, but the brain is very unique in its metabolic demands. It actually prefers glucose, and this is because it's very difficult to get others and use other sources of energy. And so another way to sort of combat that is to have the ability to make glycogen, which astrocytes can do to a very small degree. Let's look at microglia. So again, microglia are cells that help to play an immune role. They have the ability to phagocytose, right? Phagocytose means they can engulf bacteria. They can engulf dead or injured or dying cells. They can sort of just swallow them up, as other phagocytic cells can do. And so that can be another way to clear any threat, again, bacteria, viruses from the central nervous system and protect it from any oxidative stress. Now, when we think about the cells we just described, we can think about their clinical connections. So neurodegenerative diseases arise whenever we have a dysfunction of any of our glial cells. So loss of myelin, which is typically carried out by the oligodendrocytes and the Schwann cells, that can lead to multiple sclerosis. I'm not sure if anyone here has heard of multiple sclerosis, but it's pretty much a CNS disease where neurons stop working appropriately. And this can become fatal, especially when it leads to interruption of breathing, right? So when we lose the ability for the neurons that control the diaphragm to work, that can lead to paralysis of the diaphragm, and then that can become fatal. But other other manifestations of multiple sclerosis are vision problems, right? Loss of myelination of the optic nerve, which leads to vision problems. Also just weakness, motor weakness, also sensory loss, and those types of clinical symptoms can be seen with multiple sclerosis because of the loss of myelin. Alzheimer's and Parkinson's are other neurodegenerative diseases. Alzheimer's is a loss of cholinergic neurons, neurons that are releasing acetylcholine, And this is essentially replaced by scar tissue in the brain. And so Alzheimer's is going to show up with cognitive decline. So you're going to have this usually in older populations, but it can show up as a juvenile Alzheimer's. But the main presenting complaint is forgetfulness. Um, there's loss of, you pretty much unlearn everything that you were able to do. You forget how to feed yourself, how to eat, how to speak, you forget your family members, just overall cognitive decline in Alzheimer's. Parkinson's is similar. This is the loss of dopaminergic neurons, the neurons that release dopamine. And so Parkinson's usually presents with movement disorders. Dopamine helps to ensure that our movements are smooth and that they're not sort of staggered or, jittery or jagged. And so Parkinson's patient will usually have cognitive decline towards the end, but early on they present with movement disorders, very shaky when they try to move, the shuffling type of gait, and that is because of loss of dopaminergic neurons.